for a bacteria. Let's say you have an antibiotic that works on a specific protein. If they have a mutation, that protein changes shape, but still functions okay, now that drug cannot attach to that protein and cannot recognize it, they have now drug resistance. For them it's beneficial, not for us, but you're looking at the species. So that's what we mean by beneficial. Last part, so I have this chart that talks about all of these. You can look at it. Last part, again, genetics, how we have changes into genetics that are happening. I'll come back to this one. But genetics differences, humans, we do sexual reproduction. So every time an egg and a sperm fuse, you have mixing of new genes together. So you actually create a brand new human being that unless they have a twin, identical twin, their genetic makeup is unique. They're totally different than their parents and other siblings because it's just totally game of chance. What chromosome got into what egg and what sperm and so on and so forth. There are some changes in non-sexual reproduction too. You have vertical gene transfers and horizontal gene transfer. Vertical gene transfer is what you do, you give to your offspring. So when we reproduce, the eggs that I produce, I transfer those genes to my children. In a bacteria, when they split and reproduce, again, they've done vertical gene transfer. They've given it to the next generation, to the new daughter cells, new bacteria. That's vertical, right? It goes through the generations. Horizontal means I am alive, I can pass some of my characteristics to Jason, which we cannot, right? Unless you're X-Men, which actually it's the reverse. Rogue grabs somebody's characteristics in the X-Men. But we cannot, humans cannot. Most eukaryotes do not, all right? Yeast cannot do this. Fungus cannot do this. Protozoans cannot do this. Humans cannot. Bacteria, prokaryotes, however, can do horizontal gene transfer by the three methods. And all I want you to do is understand what the name means. I'm never going to ask you to explain the process to me. Just have it for a multiple choice. Transformation is some bacteria naturally are capable of soaking up or picking up fragments of DNA that they find in their nature. So another bacteria explodes, their DNA is fragmented and it's there. An E. coli that is there can have the natural ability to suck up fragments of that DNA and keep it either as a plasmid or if it has genes that are same as it is, flip them. So transformation, a bacteria picks up fragments of DNA from environment and uses it to get special characteristics. So they become transformed. Either they keep it as plasmid or they insert it in their gene. Transduction is, I didn't go over the exercise that or the experiment that told them that. It's not necessary, you don't need to know it, but I want to make sure that we finish this today without me going nuts over it. Transduction is when we have a virus make a mistake. So a bacteriophage, a virus of a bacteria comes, infects the bacteria, goes through lytic cycle. It's supposed to degrade the bacterial genome, totally destroy it and replicate. It doesn't completely destroy it, and one of the capsids, stupid thing, instead of picking up viral DNA, goes and takes the fragment of bacterial DNA. Thinks, I got my DNA inside. Packages it and then comes out. It thinks, perfect, I have a capsid, yes. I have spikes, yes. I have a DNA inside, yes, perfect, ready to go. but it's bacterial. It goes and infects the neck bacteria, thinks it's inserting its own DNA in there. Nope, sorry, it was the bacterial DNA. So a bacterial DNA, the bacteria sees it, keeps it either as a plasmid or again, kind of the same process of transformation. Here, instead of the bacteria picking it up, a crazy mistaken virus introduces bacterial genome in there rather than its own. It just packaged the wrong thing. That's transduction. So transduction, a virus introduces bacterial DNA. And finally, remember conjugation, the sex pili or conjugation pili? That's what they do. They basically connect. They take, usually just do the plasmid, take a copy of the plasmid, send it to the other bacteria. Now that bacteria has the plasmid, and they keep it. All right? All right, so don't study after this. You'll see this a lot more from here on for some areas. So biotechnology, I don't cover it, so don't study that. All right? Okay, let me go through. Oh, this one I forgot to mention. Crossing over is actually what we do to create even more varieties. So remember I said we have homologous chromosomes. One set of chromosomes we get from mom and one set of chromosomes we get from dad. Humans do something very interesting when they make their gametes, the eggs or sperms. They can come and say chromosome 1 that I got from dad and chromosome 1 that I got from mom, I'm going to swap exact pieces of the chromosomes with one another. So using the encyclopedia term. So you have book 1 that you got from mom, book 1 that you got from dad. You go and say, I'm going to take pages 1 through 300, the book of mom, and swap it with pages 1 through 300 from book of dad. Now you have books that nobody gave you. It's brand new, and you pass that on to your child. That's why every now and then, sometimes with people who intermarry, like you see it's a small island, and a lot of them keep marrying one another. They don't have to be related, but you suddenly see somebody from many generations past, they come and you're like, wow, they look alike. It's because of these gene swapping things. So we have more variety. That's why we have so many different humans in the world. All right. Okay, guys, I did go through some of the animations. Again, as you're studying, go through the animations. I just wanted to make sure that I go over chapter 8, finish everything that I need, so you guys can start studying over it, and then if you have questions throughout these next few lectures, you can ask about it. All right. Sorry, I know, your brains are fried. I actually have a slide that explains